hello to everybody, and I'm very pleased to have you all for this session of the AstroGeo Seminar. And this time it will be, we are going to biological proxy, and especially looking for a possible signature of the eccentricity with the use of coccoliths. I don't know if you say coccoliths or coccoliths, maybe coccoliths in English. Coccoliths, yeah. And this will be given by Luc Beaufort, who is at CEREGE, who has been a long time at CEREGE. He's more accustomed to recent time, but he doesn't mind going to deep time. And I think he will be interested by your reaction on his talk and the possibility given by this biological proxy for the analysis of the sediment. So he will speak on eccentricity forcing on Pleistocene carbonate production and biological evolution, which is something that was recently published in Nature. So Luc, thanks for your presence here, and you can start now and share your screen.
Thank you, Jacques, for this invitation. I'm very proud of it. And I would like to speak about eccentricity and the biological evolution. So if you are not accustomed to any idea that will be developed in this talk, don't hesitate to stop me and ask questions, because I know that it's a long time that it didn't work in the long time and cycles and things like that. So please don't hesitate to stop me. First, I would like to acknowledge my colleagues who participate to this work. Clara Bolton and Baptiste Chirac-Marx are specialists of coccolithophore like me. Antassar and Yannick Donadieu are modelers, and I will present you some climatic simulation. Yair Rosenthal is a long partner of my work. He was also the chief scientist on one of his two IODP cruises. And the work that I will present is related to two IODP cruises and images cruises in the Western Pacific and the Indian Ocean. So I will speak about eccentricity. So I give a little bit introduction on the eccentricity, not in terms of astronomy, but more on the Pleistocene time view that we have. I know that you are all working in the deep time, and eccentricity has not the same meaning for people working in the Pleistocene and pre-Pleistocene series. As we all know, eccentricity is weak now. The Earth is going around the sun almost in a circular way, but this has not been always the case. For example, the eccentricity was much stronger about 200,000 years ago. And we can see that here 200,000 years ago, we were here, the eccentricity was much larger than just now. And this could have enormous consequence on climates. And but this has less impact on insolation than other orbital parameters like obliquity and precession. Precession is modulated by eccentricity, but here we see insolation records in Hayes and in Brie in 1976, at the beginning of the rediscovering of the Milankovitch cycle. In fact, I met John Inbri during my PhD, and he told me, but why do you find 100 kiloyear cycle in your record? I was working in the Miocene, since there is almost no insolation variation on the eccentricity, on the insolation. No eccentricity. Here you see that this is a plot of insolation at 55 degrees south, and here 60 degrees north. And you see that 100 kiloyear cycle is not there. And so they were surprised. This is the same paper, one of the iconic paper on the understanding of the presence of Milankovitch cycle in deep sea sediments. And here you see the time is going this way. The ice volume variations, which follows the 100 kiloyear cycle, and the surprise that insolation is not present in this. So at that time, they were working a lot on explaining why the 100 kiloyear cycle, which is extremely prominent in the last million years, why it was there so importantly in ice and also in climate, and what it was so well recorded in the sediments. And then the idea was very early that a nonlinear response was the cause of the presence of the 100 kiloyear cycle. And here is one of the first paper by Inbri on the modeling of ice volume and the way that the fact that the time constant and the ice, when the ice has a sufficient volume, and the fact that the melting was fast and the growth was slow of the ice volume, was pushing the orbital cycle to move from high frequencies like tilt and the precession toward eccentricity mechanism. Later, Didier Paillard proposed that the nonlinear was a mix of ice volume and insolation. And so the time is also going this direction. And here it's go from the last, from 2 million years to the present. And you see that the ice volume is increasing. And at one point, the ice volume is shifting from a frequency of 40 kiloyear cycle to 100 kiloyear cycle. And it's when ice volume is sufficiently important to explain that insolation is shifted to in the eccentricity band. But yet, we see in this record that there is a 100 kiloyear cycle here, but not the 400. So the eccentricity is not fully developed in the Pleistocene. And then there are other people that say that the 100 kiloyear cycle in the last 8 deglaciation is not due to eccentricity, but in fact to obliquity. And so it's even not sure that the presence of eccentricity is in ice volume. Here we see, for example, Huber explaining that, and it was the same in Liu, showing that at each deglaciation time, they were fitting better with obliquity time and an obliquity change that we can see here than precession or eccentricity. And therefore, a change in the tilt could explain if it goes from 80, in 2 times of 82,000 years, 2 times the obliquity period, or 3 times 136, and you have this deglaciation. So the deglaciation could be, if there is a 100 kiloyear cycle, it will be a pseudo 100 kiloyear cycle due to multiple of the obliquity. So, and also Ruddiman and Remo and later many people show that this is the record of delta O18 in Izeki and Remo for the last 2 million years. And here is the evolutive periodogram, and you show that it's the 100 kiloyear cycle is only present in the last million years. And before that, here you have the 41 kiloyear cycle, which is dominating, dominating before 1 million years. So here, the earlier part of the Pleistocene, there is no there is no eccentricity. There is no eccentricity before 1 million years in the delta O18 record. The delta O18 is mainly ice volume, and it records also the global climates in a way. And so it's very intriguing that there is no 100 kiloyear cycle in this area. And also the fact that there is no 400 kiloyear cycle before is also a prominent feature. So climate in the Pleistocene has almost nothing to do or very little to do to eccentricity. And so you say, when I say climate, I should be explaining a little bit more. For example, here, in a core which has been taken in the warm pool, in which we can measure in blue here the delta weighting. So in a way, the high latitude dynamic of the ice volume. And in red, this is the SST measured by magnesium calcium in the foraminifera shells. And this, the magnesium calcium, records the SST, the sea surface temperature. And in this place, which is all the time during the year above 30 degrees now, this is the most warm area of the ocean, is where you could think intuitively that this is disconnected from high latitude ice volume. But still, you see that the red curve and the blue curves are following the same cycle. And you see at the same time here 400 kiloyear cycle in the deep part of this record, and in the younger part, it's 100 kiloyear cycle in both records. So that means that low latitudes temperature of the ocean is also following ice volume. So mainly the global climates are following obliquity, for example, in this area, but not eccentricity.
Excuse me, Luc. What is the proxy to measure the sea surface temperature?
Here is a foraminifera shell, and inside you do the chemistry and you measure the magnesium and magnesium, and you compare to calcium. And the ratio between magnesium and calcium is dependent on temperature, sea surface temperature. So when you measure this ratio of elements inside the shell, you reconstruct sea surface temperature.
And this is something that has been calibrated in laboratory?
Oh, yeah. This is used by many paleoceanographers. And this is quite, this is classical now.
And the relation with the delta oxygen 18, it has also been calibrated in a laboratory?
Oh, the delta 18 is not related directly to temperature. There is a third, which this is a benthic curve, I think, in blue here. And this is related mainly a third by temperature, a third by ice volume, and also a third by salinity. So this is a proxy of ice volume mainly, especially because we are in the benthic. And for example, here I changed my graph. Here this is a delta 18 from Lisecki and Remo, and this is a standard, and it has been used by many people to be a proxy of ice volume.
Okay.
So here you have ice volume basically in blue, and temperature, sea surface temperature at the tropic, at the equator, in the same core. So they are, I think, magnificently correlated together.
Yes, they are.
Here, not all there are climates which are not independent from global climates, but especially in low latitudes, you can find records which are related to precession. Here, for example, a primary production at the equator is often characteristic of Enso-like cycles. And you see here that there is a strong precessional signal. In red, behind is the insolation at the equator, and you see that there is a peak each time of primary production, which is related to precession. And the same with a monsoon from delta 18 in Chinese Paleotheme. So this is, you don't see, this is a work by Cheng et al. In Nature, and they produce, this is a composite of several speleotheme, and there are millions of data, maybe not million, but 1,000 of data, and also measurement of uranium thorium. So the date is independently, is not done with a relative stratigraphy like in the sediments, oceanic sediment. Here is absolute dating. And you see that all these layers in this Chinese Paleotheme, which are related to the monsoon, are directly correlated to the insolation. And when you do a periodogram of it, is a beautiful precession signal. So I don't say that climate is globally always working with delta 18 of the benthic foraminifera. There are also parameters which can show precession, but I don't know any in the Pleistocene climatic parameter which is following eccentricity, and for example, no parameters follows the 400 kiloyear cycle. But when you go into the pre-Pleistocene sediments, this is not the case. You find a 100 kiloyear cycle. This has been shown, for example, by Herbert and Fischer in 86, studying some black shell in the Mesozoic, and they found alternances between shale and limestones, which follow 100 kiloyear cycles. Here, this is the F, and here it's the B. And this was the first record. And after it has been found, we have many records showing 100 and 400 kiloyear cycle in the carbonate. Here, this is the carbonate content, so the percentage of calcium carbonate. And here we don't see the latest evidence. I think it was a magnificent work by Drury et al. In Climate of the Past just last year. And it covers the entire, the Neogene from the last 30 million years. And we don't see very well, but there is here the 400 kiloyear cycle in this periodogram, and 400,000 the 400 kiloyear cycle, and the 100 kiloyear cycle sometime. Here, there is a blow up, and I think it's magnificent, the fact that the carbonate is following very well the eccentricity. This scale is inverted. So this is important. You have more carbonate during low eccentricity period. Maybe I will have the time to come back to this, but this is not obvious because, for example, here the productivity is high during high eccentricity. Here you have low calcium carbonate in low eccentricity. So this is but I think this is very obvious that eccentricity is kind of important characteristic of carbonate content in many sediment period. Also, another place where eccentricity has been discovered is the carbon isotopes. Wang very early, maybe 15 years ago, started to publish on Pingxiang Wang started to publish papers on the presence of 400 kiloyear cycles in long delta C13 records. Here I present two in a paper from Wang, Tiang, and Lawrence, and they were showing that it was extremely prominent in series older than 1.6 million years. And this was less evident after 1.6 million years. And all except in the Mediterranean Sea, I didn't present it here. In the Mediterranean Sea, there were no, the 400 kiloyear cycle was continuing. But Pingxiang Wang writes that the mechanic behind this 400 kiloyears remain elusive, and he proposed some hypotheses. More recently, Westerhold presented a work covering the Cenozoic, a record of delta C13 and also delta O18, composite records. And here we don't see very well, but here there are some places where the 100 kiloyear cycle is present, and the 400 kiloyear cycle is almost present all the time in through the Cenozoic. So it's very, and also in some place I didn't present, but the delta O18 also had some presence of the 400 and the 100 kiloyear cycle. So it is found, as you know, because it is used in cyclostratigraphy. So by you know that the 100 and the 400 kiloyear cycle are present. But the thing that I'm astonished that it's so evident in the past, but when you look here in the present, in the last million years or the last 2 million years, it is not present. So why we have this vanishing record, and what is the cause? Most of hypotheses to explain this 100 kiloyear cycle is in all climates are related to low latitude hydrological climates, like monsoon, through erosion and weathering. Also, there are other which include oceanic productivity linked with Antarctica and silica leakage, for example, Wang. But this is very hypothetical. And in general, all these hypotheses include climatic parameters, therefore, and those climatic parameters are known to not follow the eccentricity in the last million years. So this is problematic to me. And I was always fascinated by this and wondering why we have this difference in the Pleistocene and the pre-Pleistocene eccentricity records. So here, I don't say that what I'm written here, I want to investigate the implication of non-climatic parameters such as biological evolution. It's not exactly that. In fact, I was studying the biological evolution with my colleagues and found that it was a precession in it, eccentricity record. And then I say, maybe it was working. So I'm a little bit lying in this thing. I didn't want to investigate this, but this is a good transition to my work here. So I start to explain first the nanoplankton here. And the coccolithophores are these algae. Here is Emiliania Axley. It is an algae. Here you see the chloroplast. And this is inside the cell. And so therefore it is autotrophic organism and unicellular. It produces some calcite plates here, and that you see here beautifully outside the cell to protect itself. And you have many different types of coccoliths through the time, and they are evolving rapidly. So you have a changing shape, and they are very often used for biostratigraphy. Here you have the record, for example, for the last 6 million years, and you have all the appearance and disappearance of places, which are well determined and can help to do biostratigraphy. Also, they are very important because they are abundant in the sediment, and if they are so abundant, it's because they are extremely productive. Here is a satellite imagery from the last, for about 10 years. And you see in white, here the blooms of Emiliania Axley, which can be seen is so, it is so productive that when you take, for example, here, Tara is a research vessel in France, which took some sample in this area, and I was able to look at this sample during this bloom, and you see the amount of coccolithophores is only Emiliania Axley. Here you have optical images of it, of the bloom, and here SEM image, you see the cell, and you see the free coccoliths, which are out of the cells. So in fact, this white cloud in the sea is due to the calcite produced by this type of organism. And here is a characteristic of by satellite image, you have the calcite content of the surface ocean, and you see that it mimics the coccolith image bloom. So there is a relation between carbonate in the sediment and the bloom of this coccolith. So they are producing a lot of calcite, although they are very, very small. And the group which is the most productive inside the coccolith, I will show that it represents about 50% of the mass of calcite of coccolithophore, is a group called isocresidal or Noelarhabdaceae. And here I present the most dominant the most abundant species now is Emiliania xlehi, which can be a the shape can change quite a lot depending on the part of the ocean where it is. But you have also other groups which are closely related and marked here by a bridge inside the central area of the coccolith, which is here. And depending on the side, have different names. And we have about 10 species right now of Noelarhabdaceae which are present. And in the sediments, you can find, this is images from the sediments from a core that we took a few years ago in the Whirlpool, and you see at different time, different species. And you see that here there are the different species, they are defined by the size. And here is images of a cocosphere, so the cell and the coccoliths of the same, inside the same individual, I mean by row, and different species, different rows here, and the column. And so this is one cell, which is extracted, and this is another one, but of a different species, and here a different species. And you see that there is the size is also changing inside an individual. So in fact, there is a kind of a relation between size going to small here, inside individual, and as I show, the size here is again the size of coccoliths inside the same cell and with different species, with different colors. And you see that there are some overlap in these groups, although they are different species. But genetically also, there is also overlap. Here, the different color bands. This is a DNA study of the different species of this group. And you see that there is a kind of interbreeding in a way in this group, because there are some genes which are found in a different species which are very similar. And so in fact, the species are overlapping in terms of morphology, but also in terms of genetics. But what is very interesting is that these groups are, this is the different species, and this is a genealogical tree of this group made by DNA. And you can find that they had all one ancestor, a common ancestor. All these closely related species as a common ancestor, which is dated 400,000 years ago. So all the species had evolved from one species, which were a medium-sized species about 400,000 years ago, and evolved very fast, which is very fast for biological organisms, in a different species with genetically differentiated. And so, and they did that by size, and you see that the group of species which is larger is far away from the group which is smaller, and they originated much the oldest. And then you see that distinguishing the size and genetically they are displaced. So there is a strong relation between size and size of the coccoliths and the genetics. And also we know that the size and the calcification of the coccolithophores is related to environmental parameters in this group. For example, here, this is a relation between carbonate concentration in the water where they formed and the coccolith mass, which is related also to the size. When the ocean is less acidic, they are kind of heavy and large. And when the ocean is more acidic, is very light, is like a pH of 7.9, they are smaller. Here there is a work by Bollman and Hendricks, and they shown that also the size of the Gephyrochapsa, this group, is related to the temperature, and you see the different group at different temperature. So the size is dependent on evolution, but also in ecological parameters. So this is very interesting. It seems that there is a strong plasticity in this group to evolution and adaptation to the environment. And this go through the size of the coccoliths. So we wanted to study this in the last 2 or 3 million years to see if the evolution of the size and the calcification of this group was showing a sort of a strange dynamic, or it was following climates or what. So we took 8 cores taken in the Indian Ocean and the Pacific Ocean and we took in these samples 8,000 samples to conduct this research. So this is the length in terms of time of the core that we studied. So we had only the IODP cores are going to 2 million years, but most of the Marion Dufresne images cores are shorter, of course, because we don't have it's a coring system and not a drilling system. So most of the cores are located in low latitudes. And here I present a combination of all these works. The analysis has been done using artificial intelligence to gather about 7 or 9 million measurements of coccoliths, and they are all put in these graphs. And you see that here in blue you have the average size of all the cores and all the samples together bind into 30,000 years bins. And here is a distribution of the size, and you see that, of course, the mean is going this way. But you can see that sometimes the distribution is extremely large, and there are kind of in this area, for example, you have few coccoliths, and here you have more coccoliths which are large like this, and small coccoliths like this one, which are abundant in this place. And before, 400,000 years ago, here, this is Gephyrochapsa caraebenica, which is present and dominant in this area, is the guy that I told you was the ancestor of all the Gephyrochapsa, which has evolved. We see the evolution here on the size is increasing towards the Gephyrochapsa oceanica, which is here, and toward Gephyrochapsa ericksonii, which is this small bug. And here you have Emiliania oxleri, which is a middle size, which is here, which is now dominant. So here you have the evolution of the size and you see that is going cyclically. Here you have a divergence also. Here you have a node at about 700,000 years, and then it diverges in 2 groups, a small one and a large one, and after it converged to a middle size organisms. And you can see that there is like an accordion work. The accordion here is inflating, and here is compressing, and the size is doing this strange shape. And now the size here is in blue, and in red here you have a low pass filter on the eccentricity, and you see that is kind of a mimic of it. But here I told you that it was bin data, and I was not very interesting to work. It was not very interesting to work in a bin data, and I would prefer to work on an other system which is more model of the evolution, because I told you that the average, the size, can be related to environmental parameters. So we tried to do something which was more related only to evolution. So we developed an index called Morphological Divergence Index, MDI, in order to see the evolution of the coccoliths, because here we see that there is a kind of a succession of evolution, and we wanted to shape, to follow the pattern here, but to do some time series analysis, but on 3D images is very difficult. We wanted to have an index. So here in black, this is a conceptual model. You have one species, for example, Gephyrocapsa caryabenica, which evolved in two species, Ericksonii, the small one, and the large one, Oceanica, for example. And here you have it changed to its abundance. Here the small one represents 70%, only 25% here. So the mean morphology here doesn't change, because here you have one species, which is the mean is 2, and then when it diverges in two species, since they are 50% each, you don't see any change in the mean morphology. And then here you start to see the fluctuation. So the blue curve, which is the average, is following more the ecological change, which is important on the abundance of the different species, but it's not a good evolutionary index. Instead, if you do the difference between this class and this class, and any class, here you have 2, and if you do the small minus the large, you will have 0. And then when you have this evolutionary event, and if you measure the size of this one minus the size of this one, you have an increase to 2, and it doesn't change. So the fact that you are using this type of divergence index, which is just the measure here, you have the distribution. You have one species. If you measure 2 classes, and the difference between 2 classes, you have almost 0, which is the case in this area. And in this area, you have a distribution which is bimodal. And then you measure the average here and the average here. You have something which is large, but the mean is very similar to this one. It is moving here. The mean is in black and is printed here in blue. And you see that you have a fluctuation, which are precession. And here, if you are doing just the MDI, you have something which has a different dynamic and follow more the accordion, what I was speaking about, something converging and then diverging and converging, and you see this on this. Instead, if you do the mean, you have something which is completely different and follow more the ecology. So we use this MDI as a concept to follow the in the core the evolution. Here is the different cores which are in different oceanic model. You have a place which is in the warm pool, over in the monsoon area, over in coastal area, and in all these different areas, you have different species, and sometimes you have mainly small coccoliths, in other place large coccoliths, and even with that, with the different in the different cores, we find always the same pattern, the evolutive pattern of MDI. Here you see that we have always the same, this is the 9 cores, and we have always exactly the same scale here of MDI. And in red is average of all the cores, and in black are the value from the specific cores. And you see that in all cores, we have a good pattern in every cores, which are in different oceanic mode areas. So ecology is not there. This dynamic is just forced by evolution, in fact. And this pattern is known. We know that here is the appearance of the Gephyrocapsa oceanica in this thing, in this place, we have Gephyrocapsa caryabenica, and this bloom in Gephyrocapsa caryabenica is known. And here we have the appearance of Emiliania Huxley, and this explains this shape. So we know that it's related to evolution. So MDI, in a way, for us, is evolution. And here is the stack of all the cores. And in blue is all the data in top. So it gives you an idea of the distribution of all the different cores. And here I place the eccentricity. And in the between, you have an evolutive coherency periodogram between the two curves. You see that there is a strong coherency at 400 kiloyear cycle. Here we see the 400 kiloyear cycle. Oh, sorry. And also 100 kiloyear cycle, 124 and 95. And we see the peaks here, which are every 100 kiloyear cycle. Below that, it's raw data. I mean, the resolution is 3,000 years. So
Luc,
yes.
How is established your time scale for the cores for your sampling?
It's established with delta O18, which is, as I presented before that, the LR04 is a Lisecki Remo stack of benthic foraminifera. So we have the benthic foraminifera delta O18, which is almost global. And so in a core, when you have the benthic foraminifera, you can signal, you can tune to the standard. So it's precise at about, people think 10,000 years of precision, but here we are working with 100,000 year cycle, so there is no problem of this. In a way, this this stratigraphy is well established with an accuracy which is higher than what we are speaking about here. So, but also there is kind of noise, which is a biological noise in a way. Is why this is a natural. The noise is not due to the datation, the chronology is due to the biology. So, as I told you, eccentricity is not present usually in the Pleistocene. And here you have an entire Pleistocene which is dominated by eccentricity. And we don't have, I don't present it here, but trust me, we don't have any precession or obliquity in this dynamics. After that, it was completely flat. We see here the, oh yes, it is present. Here it's the periodograms, which is placed in an awkward way, but you can see if you turn your face, and you see that in the tilt here, we don't see any tilt, any obliquity, any precession in this record. So we try to find the reason of why in a way. So we did a modeling simulation at a place, at a time where we had high, in black here is the eccentricity, in green is a precession, and we wanted to do a climatic and simulation at high eccentricity and low eccentricity, using, and at the same time, different precession configuration. So we did 8 simulation. Here, this is a climatic, the response on the wind, but we place the simulation also carbon simulation in the system, and to know the productivity, to produce the productivity. Here we have, oh, so this is to see what we did, I present this graph, which is a little bit complex. Maybe this is a present, and this is a year. We have January here, July, April, October. So here it's insolation, and we see that the insolation, it doesn't change at 0 degree right now. It is almost constant around the year. This is due to the fact that the eccentricity is almost circular. This is not a circular orbit. It's almost a circular orbit. And at the time in your simulation, in this time where simulation is very low, here we have the two simulation, the two insolation. They are in different colors, but they are on top of each other. The insolation doesn't change through the year. Instead, when eccentricity is quite high, you see that when the eccentricity increase here in blue, when the perihelion is at 87 degrees, we have higher insolation in January because the Earth is closer at that time to the sun. And in July, Earth was far from the sun, so we have less insolation. And about 10 or 12,000 years later, it is a red curve, the insolation was different. Here, precession has no effect. And here is our simulation of primary production in the Western Pacific warm pool. And we see that when the eccentricity is very low, there is no difference between the two precession simulations, and we have a weak seasonality. Instead, when the eccentricity is very large, the productivity in the warm pool was very different through the years. During the season, you have a period where, for example, here in red, when the perihelion was located in September or in August, you see that productivity was much larger in September, October, November, and in May it was very weak. We don't discuss the ecology of that, but what is important is to see that the coccolithophores and other phytoplankton which were living in the low latitudes had to face an environment which was very changing through the year. Instead, in this, when the low eccentricity, the seasonality was weak, so they have a very uniform environment. So the ecological niches are very diverse when the eccentricity is large and very homogeneous when the eccentricity is low. And this must have ecological implications and also evolutive implications. And here we show, we calculate the seasonality. What we define seasonality, in fact, is the size of this arrow between the lowest productivity to the highest, the difference between lowest and highest for a year. And we see that during eccentricity, it is very large. And here during low eccentricity is very low. And this is the result of our simulation. When we do eccentricity max, the seasonality, eccentricity max minus eccentricity minimum, we have this map. And we see that we have almost 100%, this is 100% of the yearly productivity difference in the season. That means that the increase of productivity, the difference of productivity during a season when eccentricity is high is almost the same magnitude that the yearly productivity, which is huge. This is the effect of seasonality on productivity is extreme when eccentricity is high, which is very important. And here we have all our simulation. The dots are the simulation and the primary production seasonality. And here is the MDI of the stack series, which has been sorted by eccentricity and grouped by to see something using these boxes. So here we have all the group of samples which has been taken in low eccentricity, and here the group in gray, this box related to high eccentricity. And we see that we have an increase of the MDI. The evolution is related to the seasonality. So in fact, we are thinking that evolution is related, evolution of coccolithophores is related to the niche diversity. When the seasonality is low, the niche diversity is low, and therefore the morphology diversity is low. And the reverse when seasonality starts to be high. So we explain this system related only to seasonality and not climate. Here we come back to the data from the beginning, and we see for the last million years, the variation of the morphological divergence index here for the last million years, which is follow eccentricity. And here I put back the delta O18 of the record, the pattern that we use for stratigraphy. And we see that we have only the 100 kilo-year cycle, which is maybe or not related to eccentricity, but which is very different from eccentricity. And here, this is monsoon, which is related to precession. And there is nothing related to these two major climate system in low latitude monsoon. There is no relation between this blue curve and this blue curve here. This is not possible that monsoon explain this. And this is not possible to explain this curve by the delta O18. It's much easier to explain this. But you may have noticed that there is a kind of a lag here. I will try to explain why this is shifted now. Why evolution can explain why we have a shift of this curve through this curve. There is a possible a relation. So this is maybe more complex. I'm sorry, this is a strange graph. What we know, and this is from data, and it is known since the 70s, I think it is Hans Stirstein who showed that Emiliani-Huxley High started just after 290,000 years ago. So the species is found everywhere in the ocean directly. The time is going this direction. Here, we know that all this since 270,000 years, we found Emiliani-Huxley High. But what is important is that it starts its dominance very late. Depending on the places, around 90,000 years ago, Emiliani-Huxley High start to dominate. Here is present, but not abundant. And this we think this is why we have the shift that we are speaking about. So we design a low-pass filter which try to reproduce this increase of abundance. So the blue line is the result, is a model of a low-pass filter of eccentricity. A low-pass filter, when it's designed, we can use Bott diagrams to describe it. So here, this is periods, and the magnitude of the attenuation is seen here. So the attenuation of the magnitude of the signal is dependent on the periods, and the eccentricity, precession, sorry, in blue, has no chance to be found in a system when we use a bandpass filter having a 200 years in a parameter. And here is the phase related. The phase also is shifting when we do a low pass filter. We are in fact making a lag in the system, and for 100,000 for the 400 kilo-year cycle, we have a lag of 70 kilo-years, and we induce a lag on eccentricity of 45 kilo-years, and we have a less lag for the precession and tilt, but it is erased from the series. So here is in black is the eccentricity non-filtered, and eccentricity filtered here is in red, and you see that is fit better with the size. So in fact, the fact that there is a time for the dominant species like Emiliani-Axlei, during the time that it appears and the time it starts to dominate into the ocean, it takes time, and this produces in our system, in the blue curve, in fact, a smoothing function, and this smoothing function produces the lag that we find here between the two curves. So I hope you are not still sleepy now, and I will try to wake you a little bit now with what why would I care about evolution? It's also because I told you at the beginning that the coccoliths are very important as are massive calcite producers. So the question that came is this evolutive pattern could be seen in the coccolith mass, in the mass accumulation rate of the coccoliths. And here we have the possibility, of course, to calculate the mass accumulation rate of all the coccoliths what we have, because we have the numbers, we have the time, we have the number of coccoliths, and we have the mass of each coccolith. So multiplying the mass by the number of them, we have the quantity inside the sediment. And then we know the time of due to sedimentation rate. Then you can come back to accumulation rate, which is the important parameters. So doing that, we have this series in the bottom of the graphs here. And you see that we have, this is all the graph together, all the 9 cores together. And so we have large variations because ecology there is playing a role in place where we have a lot of producers, then we have more coccoliths in the sediments. And when in oligotrophic area, the accumulation rate is lower, which is logical. But the average of the two give a pattern, although it's much different depending on different groups, different sites. We can see that there is a kind of a long pattern like this, which is a 400 kiloyear cycle, and also very well pronounced the 100 kiloyear cycle here. And when we do the coherency graph between the evolutionary pattern and the accumulation mass, which is here, we found that there is a high coherency between the two curves. They are independent. These two records are completely independent because they are not at all made on the same metrics. And still, we see very well the 100 kiloyear cycle through the entire Pleistocene and the 400 kiloyear cycle. And so this was a kind of very intriguing because the curves don't match each other. You see, you don't feel that they are similar, but the dynamic is similar. It's because the dynamic is complex between the two. And I will try to explain now this. In the top, you have the same curves than the bottom one here. In the top, in yellow, you have the mass accumulation rate of the Noël-Arabidacea that we call NOMAR, for no mass accumulation rate. And in blue here, we have the flux, the number of coccoliths of this group. And in red here, we have the size or the mass. So in fact, this curve results from the multiplication by the red and the blue. And we see that they are mirrored of each other, is why there is a complex relation between evolution and this is because in fact, the most productive coccolithophores are the smallest. The small coccoliths here are few, No, sorry, the mass here is in red. So the small coccoliths here are producing much more. This is Emiliani-Axley, which is this is a gray band here. Emiliani-Axley start to produce a lot of coccoliths, although Emiliani-Axley it's light species. Here we have Caryabenica, the ancestor of this one, which is also a small species, but produce even more. This is a bloom of Caryabenica, which is known since the 60s. And this bloom is very prominent. So in fact, the species which are small are producing a lot. And these gray bands are stratigraphically known acmes of, that means extreme production of Gephyrocapsa in the Pleistocene. They are used stratigraphically. And so that means that the small species or the middle-sized species are producing much more. And why that? And so we have an hypothesis which is related to the environment that we simulated, and I show you previously. The environment during low eccentricity period show low diversity, and they confers an opportunistic character to the species. That means that the species are opportunistic and they are more able, because the environment is less diverse, they are more able to be producive everywhere in the ocean, and they start to invade the ocean and produce a lot. So this opportunistic character makes them dominant. And it's why we believe there is this relation between evolution here and mass accumulation. So knowing that, for example, here that coccolithophores are the dominant, here is a work by C. and Rosenthal in 2020, and they show that in gray here are the coccoliths, the Mar coccolith and the Mar foraminifera. And you see that for the last 15 million years, coccolithophores are always dominant in terms of mass. So when you take a core in the center of the ocean and you have carbonate, most of the carbonate is produced by coccolithophores and very little by foraminifera. And also the Noella rhabdaceae that I study here represent 50% of, in terms of mass, of the sediment of the coccolithophores, the entire coccolithophore assemblages. This is not number, this is mass. And we are able to do all the groups of coccolithophores, and we see that it represents half of it. So half of this grape is related to the thing that I told you about this. So this explains why we think that this evolutionary pattern can drive a percentage of carbonate in the sediments. So I told you that it was carbonate, which was very important in the pre-Pleistocene thing. Another was delta C13. So this is a work in progress. I just have one graph. This is a core in Lord O Rise in South Pacific. So this is not part of the paper that I speak about since then. This is a work with Laurence Vidal and myself and a few others from CEREGE. And we compare here the MDI in red. So the MDI is a morphological evolutionary things of the coccolithophores. And in blue is a delta C13 of the fine fraction, which is mainly coccolithophores. And we see that the delta C13 and the evolutionary pattern are completely related in the same core. And we have now a master student who is working also in another core and found also the same pattern. That means a relation between the fine fraction delta C13 and evolutionary pattern of coccolithophores. So this is, I don't know exactly all the implication of this, but there is a relation between the delta C13 of coccolithophore fraction and evolution. We don't know exactly why, certainly with vital effect on the coccolithophores on delta O18, delta C13, and this is the reason why we think that carbonate and delta C13 are influenced by coccolithophores. So as a summary, I would like to stress, I think I shown you that eccentricity does not have an obvious effect on Pleistocene climates. But eccentricity, as many people have shown, gives the tempo of oceanic carbon cycles in the pre-Pleistocene sediments, and therefore is why you are using in cyclostratigraphy. But the question is, where does it come from? And here our explanation is that coccolithophore evolution follow eccentricity in the Pleistocene when climate does not. And therefore, and coccolith flux, coccolith and coccolithophores are different, obviously. Coccolith fluxes are related to coccolithophores evolution and follow also eccentricity. And since coccoliths are dominant producers of pelagic carbonate, it is logical to think that the percentage of carbonate is related to the coccolith and therefore to the coccolith to the coccolithophore evolution, and it's why we have the eccentricity in pelagic carbonate. So our tentative answer to the question is maybe evolution of coccolithophores and other phytoplanktons, because we are almost certain that not only coccolithophores are following this evolution pattern, but diatoms, all organisms, even maybe not phytoplankton, but other planktons living in low latitudes, because the diversity of the niches will be different, will evolve following the eccentricity. And all of this conducts that the carbon system orchestra, the evolution conducts the carbon system orchestra, has been by providing the tempo of the eccentricity bits. Thank you.
Okay, thanks a lot, Luc. And we are now open for the question. At the end, there is something I don't understand, is the difference between coccolith and coccolithophore. Can you?
Yes, very easy. A coccolithophores is this, and the coccolith is this. In fact, a coccolith is a plate, and the coccolithophore is the organism which produces the plate. So, in fact, a coccolithophores produce coccoliths. Foreign mean.
But I don't. You say the coccolith is one single piece.
Plate. Yes.
And the coccolithophore is the aggregation of these plates.
Yeah, this is a cell which live inside, in fact.
Oh, there is
the coccolithophores is the organism, and it's like we'll say coccolithophore is the equivalent of a human being, and the coccolith is the equivalent of a bone of the human being.
Okay. And it produces, and the number of coccolith evolve in time?
Depending on species, and also it evolves during the day. For example, there are more coccoliths at the end of the day than at the beginning of the day, because they are produced every 1 hour, and they go out of the cell to produce this. So you have, for example, this Emiliani-Axley here has been taken in the morning and has about 7 to 8 coccoliths, and at the end of the day it will have 12 to 14 coccoliths. And it will divide during the night in 2 other cells, having half of the number of coccoliths. And some coccolithophores species have more species than the other, depending on the size and the shape. But Emiliani-Axley is a high producer. There are some coccolithophores which are large, which produce at a rate of several hours. Coccoliths, and they stop calcifying after a few days.
Okay, so any other question? If there are, I have some other one. So here you explain the time lag, which is quite clear. The time lag is due to the evolution of the time it takes for the specific species to become dominant, so that its effect is visible in the sediment.
Yes.
Yeah, and so this is quite clear. So this time lag is quite big, in fact.
Yes.
Quite
And
large.
This should have, if I'm right, if my hypothesis is right, then this makes for you a big problem, because you have to infer this 70,000 years lag into your reconstitution of cyclostratigraphy.
Yeah, but all the proxy, because you assume that all the proxy that are seen are due to the coccolite?
If carbonate, if you use a carbonate system, maybe.
If you use a carbonate, something that is related to carbonate, what you are saying is that most of the carbonate comes from the coccolite and not from the forams. And so your mechanism applies to all proxies that come from carbonate. Is that what you mean?
Yes, in a way, if you are looking in
from organic carbonate, because there is also the precipitation carbonate, no?
No, this is very rare, only in septa. It's in a very, it doesn't exist too much. Most of the carbonate can be aerated from platforms. They can arrive from coral erosions, or even can be inherited from the sediment, arriving laterally. But also, most of it is a pelagic carbonate when you are looking at the pelagic core. And it's mainly coccolithophores, maybe 30% to 50% foraminifera. But maybe foraminifera also are evolving following the same pattern that I see. And maybe also everything is evolving at this place in low latitudes. It is because the coccolithophores have a very well, I'm sure this will be found in other groups in a way, when this will be studied. But okay. It's not sure that I'm right, and maybe it may be monsoon. This, for example, here's this paper by Anna Joy Drury and others. You have this relation of carbonate percentage here in red and eccentricity, and they are related, obviously. And my position here is that coccolithophores in this site is produced mainly by this carbonate here is mainly coccolites. And here this percentage is reverted. So it's exactly what I say. Here you have a low eccentricity, then you have a high production of coccolites related certainly to Anola rhabdaceae and other coccolites which are producing a lot. This is my point. And in that case, this should be shifted by 75. This date here, which is calculated on carbonate here directly, and is fitted directly without any lag with eccentricity, should be shifted, should be a little bit older. But this is my suggestion, and I'm sure you will not admit it, but I push it like yesterday in our presidential debate. I will push my points, but this is just hypothetically an hypothesis.
Okay, any reaction? I see one hand.
Yes, I'm
Can you
Marina?
Yes.
Marina Rabino from Brest. I'm sedimentologist in Brest, and I had a question to Luc because if I understood everything correctly, you see a big change around let's say the last 400 kilo year, and without a clear eccentricity. So I understood that. And I was talking not so long ago with Andre Droxler, who's working on carbonates and reef platform carbonate. He also sees that big change in his reef construction. So I was wondering if you, like, if those two are matched, and that's something is happening in carbonate global concentration on the ocean or something.
This is a very interesting question, because there are some papers showing, trying to see a shift of carbonate producing system between platforms and pelagic sediment and pelagic ocean. And this was Brad Updike was the kind of the promoter 20 years ago. But the fact that it was a kind of a decoupling between carbonate reefs and the pelagic carbonate productions. And this will cut at Isberger at the beginning in the 70s, who proposed even that the glacial interglacial variations were related to the fact that when the sea level was lowering, the carbonate producers were not anymore in the shelf because the shelves were exonded, and the carbonate was going towards the pelagic system, where then the coccolithor 4 would have been more producers. And I know that Andre is working a lot on this, but it's more difficult to compare this type of results with carbonate, because they don't have shelf carbonates. They don't have records of production as precise as we have here. So I'm waiting Andre to give me a good curve, and we will be able to work together to see if they are working in phase or out of phase of what we show. But this is as truly a very important application for us.
Thank you.
Is there any? Yeah, but this offset is somewhat annoying, and as from what you say, it will also be in the carbon content, in the all carbon indicators. Is that what you say?
Not necessarily organic carbon, but calcium carbonate. In the carbon from the calcium carbonate, yes, is a carbonate system in the ocean, which is will be influenced in dramatically by this. And so everything which is based on.
Didn't you show that the carbon content was also following?
I speak about delta C13 because delta C13 is an other compartment of the carbon, which is where the delta C, the eccentricity has been found in pre-Pleistocene climates, pre-Pleistocene sediments. And here we see a relation between carbon, delta C13, so this is carbon, and we don't know why we have such a good correlation between MDI evolution and delta C13, but we have it. And this indicates that coccolithophores also are influencing the carbonate part of the calcium carbonate. And therefore, this is carbon for us.
I have a question. This is Paul. I can't raise my hand. I'm in my car.
Yeah, you're fine.
My question.
Try to keep an eye on the road.
Yes, I do. Is there a role for nutrients? I mean, you can't have more coccoliths than the nutrients allow, and the nutrients are controlled by the, in part by the climate system. So I'm wondering if there is some role for nutrients in the cyclicity, and it would be great to see what diatoms and radiolarians are doing at the same time.
Yes, Paul, this is an excellent question. And I don't know how to answer this because we just published that a few months ago, and definitely we want to study the diatoms and the radiolarians. I have a student who is No, he's not a student anymore. He is a researcher now. A former student who is doing radial Arians flux is in the same core than we are doing. And the data are very interesting. The diatoms, they don't preserve very well in low latitudes where we are working, but we would really love to have diatom results. Organic carbon is a good thing. A problem that I have is that productivity, for example, you don't see the curve, but I published 25 years ago a paper showing that productivity in the area that I discussed today was changing through precession. And unfortunately, the time where the productivity was highest was when the eccentricity was the highest. That means that productivity was highest when the carbonate production was the lowest. So I cannot give you a clear answer, but you are right. It should be correlated, but our first data, in fact, show an anti-correlation. And this is annoying, I know, but this is, I cannot tell you more about that, but I definitely work on that now, and maybe I hope others will work also in the same thing to find new rules and new evidence on this. But no, I cannot answer completely to your question yet. But it's a thing that we are thinking about, definitely.
Any other question? I don't see some. So thanks, Luc, again for this interesting and puzzling story, because if we need to put such a big offset, it's a problem in all the proxy. I don't know if what is the outcome.
Can I say something?
Yeah, sure.
The lag is the weakest part, I think, of this story. If maybe it could work without the lag also. We will have the same, is a thing that we noticed. It's an observation that we have only for the last 200,000 years, but it may be not. We don't need a lag always to explain the MDI. The MDI is more a prominent feature and more solid feature than the lag. But I was interested. I wanted to show that to you because I think it will be interesting for what you do.
Sure. Okay, so that's the end. Thanks for all. The next talk will be on osmium rhenium dating, and it will be given by Holstein and Judith Anna. I have not a confirmed date yet, but it will be at the end of May. We have to finalize the date yet.